Okay, so we've gone through our B cell proliferation and differentiation. Okay, so we know that the B cell becomes activated. It internalizes, or it binds to the antigen. It internalizes that antigen through an endocytosis process. Okay, it differentiates into our B effector cell, which is our plasma cell, as well as our memory. And that plasma cell will release antibodies, and we know now what those antibodies will do. Okay? So this is why I don't like how your book presents it, because none of that happens until we do what we're about to talk about right now. Okay? So what we're going to do now is we're going to look at the proliferation and differentiation process of our cellular immune response. Okay? And in this case, when we talk about our cellular immune response, remember, this is our T cell population, our T lymphocyte population. Okay? So T cells will provide a defense against intracellular antigens. Okay? Some of your T cells will kill directly, while others will be involved as activators of the immune response. Okay? So when we look at the cell-mediated immune response, we have two populations of T cells. We have our CD4 cells, which will become our T helper cells. And we have our CD8 cells, which will become our cytotoxic T cells. Now, you'll hear me talk about these as T helper cells, or T cytotoxic cells, or cytotoxic T cells, or helper T cells, okay? But I typically will refer to them as T helper and T cytotoxic, just because the TH and the TC is how I write it, so that's just in my mind how it gets said, okay? So these cells here are our naive cells. So our CD4 and our CD8, okay, these are naive. So when they graduate from boot camp, right, they seed our secondary lymphoid organs as CD4 and CD8. Okay. Once they become activated, they become a T helper and a T cytotoxic, respectively. Okay. Now, the T helper is going to do just that. It is going to help our immune system. It's going to activate our B cells. It's going to activate other T cells. It's going to activate macrophages. It is going to be the director of our adaptive immune response. Okay. Cytotoxic T cells, okay, are going to destroy cells harboring foreign antigens. Okay, so if a pathogen enters into a cell, this cell is going to destroy that host cell. Okay, so the key to cell-mediated activation is an antigen-presenting cell. Okay, so a T cell cannot respond to an antigen by itself. So it doesn't bind an antigen that's free-floating. It doesn't bind antigens that are just cruising around out there. Okay, it has to be activated, okay, through fragments or antigens that are present on the surface of an antigen-presenting cell. Okay, so antigen presentation is vital for this activation of this naive T cell. Okay, and this activation requires a two-step process. Okay, so if we have, here's our antigen presenting cell. Okay, remember we have our MHC. This is our MHC molecule that tells the immune system that this is self. Okay. However, in this case, here is our foreign antigen. Okay. Now, we also have our T cell. Just kidding. This is going to be our CD4 cell. Or CD8. Okay. And what's going to happen is this CD4 cell is going to have to bind to this antigen. It doesn't telescope out, it comes to it, but I can't have it come to it on my flat screen. Okay, so this is our antigen binding. Okay, but it also has to have another kind of safeguard in that it's going to basically really put out a protein and going to verify with the APC that this is really what we want to do. So this is our co-stimulation. Okay, it has to have both of these occur because this activation is so powerful. We need to make sure that this is really what we want to do. Okay, so both of this is going to occur on the surface of the same antigen presenting cell. Okay, and this is required for that clonal selection. Okay, just like we talked about in the B cell, although it's going to be a little bit different. Okay, but similar process. Okay, so here now we can see it in real animated or real cartoons that are actually your book creates. Okay, so here is our antigen presenting cell, phagocytic cell. Whether it's a dendritic cell, whether it is a macrophage, right? Through phagocytosis, it's going to engulf that bacterial antigen. It's going to chew it up through the phagolysosome, okay? And it's going to basically present that antigen on the surface of its MHC molecule. Now again, remember, it has to be an MHC molecule because that MHC molecule tells us that we are self, okay? Now, what happens? Our T cell, our CD4 cell comes along, okay? We're a CD4 cell, okay? And what it does is it binds to that antigen and it co-stimulates. Okay. Now, again, you need both of these things. You have to have antigen binding and co-stimulation in order for this CD4 cell to become activated, to proliferate and differentiate and create T helper cells, which are our effector cell, and memory CD4 cells. Okay. Without co-stimulation, you get something called anergy. And what anergy is an inability to divide. Okay. So if you only get one of these, you get stuck here. You can't do the clonal selection or clonal formation. Okay. Okay. So we have an activated T helper cell. Okay. Activated by the antigen presenting cell. Now that T helper cell is going to go out and it's going to help the rest of the immune system. Okay. That T helper cell is going to go activate our B cell, which we already talked about. It's going to go activate the T cytotoxic cell or the CD8 cell. Okay. It will trigger that proliferation and differentiation process in both of those cell populations. Okay. So without our T helper cell, and I can't emphasize this enough, you guys, without the T helper cell, you do not get an immune response. Because again, and we'll show this here on the next slide, that T helper has to become activated. So our CD4 has to become a T helper. That T helper will activate our B cell, which will also activate our CD8 cell. This will become a T cytotoxic cell. This will become a plasma cell. This will produce antibodies. This will kill the cells of our body. As you can see here, without this, you don't get this, and you don't get this. Okay? Okay. Now, again, this is why I don't like how your book presents it, because we talked about this whole process. And at no point does your book say, oh, by the way, none of this can actually happen unless that T helper cell gets activated. Okay. This is why I do it a little bit different in the drawing that's available as a recording in this week's chapter. Okay. So let's look and see how that now T helper cell actually activates both the B cell and the CD8 cell. Okay. So here is our B cell. Okay. We talked about the fact that the B cell is an antigen presenting cell, and that is because, remember, the B cell can bind that antigen. Okay. Now, through an endocytosis process, it's going to internalize that antigen. However, it stalls right there. It can't do anything else, because what it needs is this T cell, okay, to bind to this antigen. Okay. The binding here triggers the release of chemicals that then triggers the activation or completes the activation of this B cell. So now this B cell can go and become a plasma cell or a memory cell and produce antibodies. Okay, so without this T cell there, this doesn't occur. Okay, now, in addition to activating our B cell, the CD4 or the T helper cell, now that it's activated, will also activate our CD8 cell. Okay, so on that same antigen presenting cell, okay, here we saw this T helper cell activation. Okay, once that CD4 cell becomes activated, okay, it causes this dendritic cell, okay, to express a co-stimulator molecule to our CD8. So even though our T helper cell has a co-stimulator that activates it, okay, that's not available to the CD8 until the T helper cell becomes activated. So once our T helper cell becomes activated, this sends chemicals down to our CD8 cell. Okay, our CD8 cell now has a co-stimulator, okay, that it can bind to, and that co-stimulation in combination with the antigen binding, okay, activates our CD8 cell into a T cytotoxic cell. Now, the T cytotoxic cell directly attacks and kills cells of our body that have been infected. Okay, so this T cytotoxic cell will circulate in our blood and our lymph organs, searching for cells of our body displaying antigens that they recognize. Okay, they will bind to a self-non-self complex. Okay, now they only destroy infected cells and abnormal cells, and they do this by a kind of a really cool process. So essentially what they do is when they find a cell that's been infected, okay, they release these molecules known as perforins, which are a protein, and a group of enzymes called granzymes via exocytosis, right? So it releases those molecules from vesicles within the T cytotoxic cell. Now the perforins are going to perforate. They're going to form pores in the cell that then allows the granzymes to enter into that cell. And the granzymes then elicit an apoptotic response. So let's see how this works. Okay. So this is actually its T cytotoxic cell attached to a cancer cell within the body. And essentially what's happening is the following. So here's our T cytotoxic cell. Here's our vesicle, our granules full of our granzymes. And so what happens when this binding occurs, okay, these proteins, the perforin proteins, which are here, so our perforins are these rods, and the granzymes are the little circles, okay. The perforins get inserted into the wall and form these pores, and then the granzymes enter behind it and that elicits an apoptotic response. So cool. Okay. Now, this may seem very familiar to you, where we have a cell that targets a cell that's been infected and kills it. Okay. We have, remember, our two arms. We have our innate and we have not innated. Come on. There we go. We have on our innate arm and our adaptive. Okay. Here we have our natural killer cell. Here we have our T cytotoxic cell. Okay. This does it non-specifically, i.e. it doesn't care what the antigen is. This does it specifically. Now, their mechanisms of action are a little bit different, but essentially both's goal are to kill cells that have become infected. Okay? Okay, so that completes our cellular immune response.